All right. Okay. All right, so I'm going to review a little bit today before we go into new stuff. Just so make sure we're all on the same page from last class. So we finished up the photosynthesis chapter, and we started on cell division, and we talked about something called mitosis. So mitosis is the process of cells splitting in two to make two new cells. The goal of mitosis was to create identical cells. So you wanted to take the cell that you started with and make two copies of it, right? So we ended class last week talking about the four main stages of mitosis. So we had prophase, metaphase, anaphase, and telophase. So prophase, you had condensation of the chromosomes, you had your nucleus disappear, you had the mitotic spindle form, the chromosomes would gradually attach to the microtubules and be pulled to the center of the cell. When the chromosomes were in the center of a cell, that stage was called metaphase, the second one. Once the chromosomes start splitting, which means the sister chromatids pull apart from each other and head towards opposite ends of the cell, that's called anaphase. And then telophase, those chromosomes start unraveling, the nucleus reforms, and you get cytokinesis, which is separation of the cytoplasm, right? So that all sound familiar? Prophase, metaphase, anaphase, and telophase. Things I want you to be able to know by the time we have our next test. You should know the order that they occur in. So you should know prophase, followed by metaphase, followed by anaphase, followed by telophase. And you should have a general idea of what happens in them. So prophase, chromosomes condense. Anaphase, or metaphase, chromosomes line up along the middle of the cell. Anaphase, sister chromatids separate. Telophase, you get separation of the cytoplasm. Cytokinesis typically occurs at the same time. So things like that you should know by then. And you should be able to identify stages. So if I showed you a picture like this, what stage would that be in? Okay, so why would you say telophase? Okay, you're looking at, so you're saying the chromosomes are pretty separated and you're seeing that line form in the middle. So yeah, you could say telophase. You might also say anaphase. This would be a really bad one for me to give you on a test, right? But if I gave you this on a test, I would probably accept either anaphase or telophase because it's probably right between the two. So cells don't always fit into one or the other. On a test, I'd give you ones that are really clear about them. So anaphase, you wouldn't see that line in the middle. And I think without that line in the middle of this picture, that would more likely be anaphase than telophase. Right? Okay. So review that. This should be really easy. You should just be able to say, I think this phase is here and this is why. Right? Okay, there's a few more things. These are slides I didn't give you, so they're not going to be something that's on your test, but I think they're pretty interesting. So kind of throwing in just a few more slides before we head into meiosis. If you remember back, hopefully you do, to earlier in the lecture on Thursday, I talked about the cell cycle. I talked about interphase and how it has growth phases, G phases, and S phase. Do you remember what happens during S phase of the cell cycle? It's a question you should be able to answer. What was the question? What happens during S phase of the cell cycle? Okay. Replication of what? Yeah, chromosomes. So your DNA has to be replicated. Okay. So S phase is DNA replication. It's right up there too. So DNA replications occurs during S phase. G phases, you have growth phase. These are all part of interphase, right? During interphase, and even during mitosis as well, there are checkpoints, and these checkpoints ensure that a cell will complete the cycle properly, that they're not going to have something wrong happen to them. And if you take 1010, you'll learn a little bit more about what's happening during the cell cycle. We spend a little more time talking about this. But things that the cell determines during those checkpoints are things like, am I large enough to divide again? Am I getting a signal to divide again? Or am I getting a signal that's telling me not to divide? At the end of S phase, the checkpoint will determine if DNA was copied correctly. So in order to copy your chromosomes, to make a duplicate copy of them, you need to replicate all the DNA that's inside the cell. And if it doesn't do that properly, if they made mistakes, it doesn't want to go ahead and divide. Because if it divides, you then have two cells that have errors in them. So there are checkpoints throughout the life of the cell. What do you think happens if one of these checkpoints stops functioning properly? Okay, it's not going to divide right. So it'll either divide with the DNA being damaged, or it will divide when it's getting a signal to not divide. Do you know what that's called when a cell is doing something inappropriately? So what if a cell starts dividing over and over and over and over again when it's being told not to? You know what we call that in medicine? I'm sure you've all known someone that has had this problem. You heard of cancer before? So cancer is a breakdown in this thing. And if you get a cell that divides over and over and over and over and over again, that's called a tumor. Cells that are growing out of control, you get a mass that is dividing when it's not supposed to divide. You can get benign tumors, which basically what a benign tumor is that it shuts down. So it does this a few times, and then the cell figures it out, or your body figures it out and stops the division, and so you have a lump, but it's not actively growing. And you have malignant tumors that just continue dividing, and they actually can break off, and those cells can travel elsewhere in your body and just start continuing dividing because they're not responding to normal cell signals, so they're going to be growing out of control. So not only is cell division really important, so mitosis, the ability to copy your cells, make exact copies, very important, but control of that process is very important. So if some reason your cells get copied incorrectly, they can lead to cells that grow out of control, that are malignant, that start spreading elsewhere. You can have benign tumors that are they can either stop growing or they're contained. They're in one location, and they can actually usually be removed pretty easily if they're in a not too inaccessible a place. So cancer is a catch-all term for cells behaving inappropriately, basically, which is why it's something that's very hard to fight in medicine, because there are so many different kinds. Basically, any cell in your body could become a cancerous cell if it gets programmed incorrectly and it starts doing things incorrectly and acting in ways it's not supposed to. So that's why we usually refer to cancers by their location. So breast cancer, lung cancer, mouth cancer, things like that. So skin cancer, you have cancer of a certain type of cell. But there actually could be multiple things that are wrong with them. So that's something you'd learn more about in 1010. So I'm not going to focus on that aspect of it, but I think that's probably one of the more interesting aspects of this chapter. Okay. Again, I added in a few extra slides just to kind of increase the information that I'm giving you today, but everything that you're going to need for the next test is in the slides that I put in your handouts. So the next part of this chapter, and really the second half of this chapter 8, is all about reproduction, but it's about really making reproductive cells. And reproduction in biological organisms comes in two flavors, two varieties. There's asexual reproduction and there's sexual reproduction. And really what we've just talked about and what we talked about on Thursday is a mechanism for asexual reproduction. So organisms that reproduce asexually, so that's with A is without sex, will just copy their genes and they'll split. So they'll basically do mitosis. And in bacteria, this is called binary fission. They will just copy all their DNA and then they'll split in two, and it's basically mitosis. So your cells are asexually reproducing to make your body. They are making copies of themselves. Sexual reproduction, on the other hand, involves two parents contributing genetic material that is combined in the offspring. And that's what you are the product of. You're the product of sexual reproduction. You have two parents, each of which gave a little bit of the genetic information that made you. So in asexual organisms, the parent and the offspring will be identical. They have all the same parts. And there are a lot of organisms that reproduce this way. Some of them will reproduce by budding. Some of them will just break off parts of them. So this would be equivalent to if I cut off my arm and then my arm would grow into a new person. Right? That would be a form of asexual reproduction. There are organisms that can do that. So sea stars, starfish, you can cut them in half and they'll grow each half will grow new limbs. So you have two from one. This is actually, what is that? I think it was urchins. That urchins were kind of devastating some of the fishing grounds that fishermen in, I forget exactly what part of the world this was, but the fishermen, when they caught them in their nets, they would cut them up and throw them back, figuring they had killed them. And what they were actually doing was just increasing the number of these organisms out there. So they were unwittingly leading to a bloom in number of this organism they were cutting up. So some organisms can reproduce that way. We can't. Really, there are advantages and disadvantages to both. And just, I think this is kind of an interesting topic to it. There's actually a lot of advantages. We're very biased towards sexual reproduction. We say, because we reproduce that way, we think it must be the best way of doing things. But asexual reproduction has a lot of advantages. It doesn't require a mate, so one organism can reproduce on their own. So organisms that don't live in huge colonies, that live pretty spread out, it's a very good way of doing it. It also allows them to preserve desirable genetic traits. So let's say you are the pinnacle of your species, that you have everything that's right with you. By asexually reproducing, you can ensure that all your offspring have those same traits. Right? Disadvantage is that it preserves undesirable traits, and it leads to very little variation in the offspring. All your offspring are going to look like you, and if you have defects, they're going to have them as well. Right? So this is one of the reasons why sexual reproduction exists. So, and again, this is not something I'm going to test you on, but kind of leads us right into the next aspect of this. So sexual reproduction, which what we do is, it leads to a variety of offspring with unique genetic traits. And this is something we're going to talk about once I get into Chapter 9 and we start looking at genetics a lot. We're going to look at how traits can vary in offspring. So traits that are present in your parents might be different in the offspring. So you probably know this. You probably look at your parents and you might have some traits from your mother, you might have some traits from your father, you might even have some traits that neither of them showed, and we're going to talk about why that is in the genetics chapter. So you can have a variety of unique genetic traits, and if you look at your brothers and sisters, if you have any, they're probably different than you. So you will have a unique combination of your parents' traits than they do. And those might be advantageous. So you might actually be better off than both of your parents because you could get desirable traits from both of them. However, and this is probably what you see in your siblings, right, is that they got all the traits that were not desirable, right? So they got all the bad stuff from your parents, and that's just as likely as well. So it doesn't always preserve desirable traits. It could be lost in a generation. could be lost in an individual. And so things could get worse and worse generation after generation, especially if one individual had lots of desirable traits. Crossbreeding with a less desirable individual is going to just make their offspring worse off. Right? But this kind of bottom part shows that it allows you to survive ecological disasters. So asexual reproduction leads to the same individual over and over and over again. Something changes in your environment, it could easily wipe out that species. Sexual reproduction leads to variety, and maybe one of those variations is going to survive. So we're going to eventually start talking about evolution and speciation after the next test, and we'll talk a little more about this. But I thought it'd be good to introduce now. So, humans, which we're probably going to focus on because that's what we know most about right now, reproduce sexually. We have some genetic information from either both of our parents. So we need to know a little bit more about chromosomes. We're not going to spend a lot of time talking about DNA and protein synthesis and things like that, because we just don't have enough time in the semester with all the other topics we want to cover this semester. But the one thing I want you to know is that DNA, so the genetic material that's found in your nucleus, is organized into chromosomes, and you have 23 pairs of chromosomes in humans called homologous chromosomes. The reason why we have pairs of chromosomes and not just 46 chromosomes, we have 23 pairs, is that we get half of them from each of our parents. So you got 23 chromosomes from your mother. You also got 23 chromosomes from your father. The word homologous means that they have similar origins, basically, but it means they have similarities to them. They share similar characteristics. And what they share is that they contain genes for similar traits. And we're going to get into more of this when we get to Chapter 9. But what I mean by that is, let's say you have a gene, which is a region of DNA on one of your chromosomes that determines eye color. And so you're going to have two chromosomes, each with a gene that determines eye color on them. You might have gotten a gene that causes brown eyes from your father, and you might have gotten a gene that causes blue eyes from your mother. You got a gene for eye color from both parents, but you might have gotten two different versions of it. And we'll talk about what color eyes you're going to have when we get to Chapter 9. But for now, just know that you get one copy of each of these genes from each of your parents, and they fall on homologous chromosomes. So the eye color gene might be on chromosome 1. It would be on chromosome 1 from each of your parents. And maybe you have another gene that determines hair color. And you might have gotten brown hair from your father, but blonde hair from your mother. And again, we'll talk about the next chapter what hair color that means you'll have, but you get one copy from each parent. Okay. So those are homologous chromosomes. So what do you think is the advantage, because there is an advantage, to having pairs of chromosomes, to not just having one of each, but to having things like two eye color genes, two hair color genes? Like, maybe not for those specific genes, but why would that be an advantage to an organism?
Evolution.
What do you mean by that?
Like, as time goes on, each offspring gets a dominance trait, and as it goes on, the better, I don't know how to explain. Say the taller, stronger ones are always going to be more dominant. So as time goes on, humans get better. We evolve and keep getting better because the better traits keep moving on.
Okay, all right. You're hitting some of the points, and yeah, we'll kind of clarify some of that as we go along. But you're right. It's basically, you could say, I could take out of that you're saying there's variety, and you're giving variety to your offspring, and maybe one of those is going to be better. Maybe having blue eyes is going to be an advantage sometime. Maybe having brown eyes is going to be an advantage, and because you have one of each, there's a chance you could pass on either of those to your offspring and give them an advantage, and they might survive, right? Okay, so that's part of it, is that it allows for variety in your offspring, and that variety could lead to better survival. Okay, so that's perfect. That's one good kind of benefit to having pairs of chromosomes, is that you don't pass on the same thing to all of your children. Any other ideas of why having pairs, having two copies, could be a good thing? Okay. Yeah, I mean, that's very much on the same line, which is, yeah, that's definitely good. But there's kind of, I'm playing guess what I'm thinking kind of thing, which is not very fair. But along those same lines, let me give you kind of an example. Let's say the gene is for an enzyme that helps you break down lactose. And why would it be advantageous that you have that gene on two separate chromosomes? Let's say you have a child with someone who's lactose intolerant. Why might it be an advantage to your child that they get a chromosome from each of you with a gene for that enzyme?
They have a 50-50 shot of having a dominant trait that they're not lactose intolerant. So they have like
Okay, be careful with the way you're phrasing things. We'll fix that next. But so you're saying
they have a chance of not coming out.
Lactose intolerant, right? So they're getting, what are they getting from your partner, you think? Are they getting, so they're getting the gene that maybe doesn't work, that isn't able to break down lactose, right? What are they getting from you? One that is able to. So you got one defective gene and you got one working gene. So by having a pair, you now are more likely to turn out not lactose intolerant, right? So we'll talk about in the next chapter that it's possible that you are able to break down lactose and then you pass on that defective gene to your offspring. We'll talk about dominance and things like that. But that's the other half of this, is that in addition to creating diversity, which is that second part there, it also is compensation for defective genes. So one chromosome may have a functional version that compensates for a defective version on, that should be defective one, not defective on the other chromosome. So pairs really helps balance things out. So you don't necessarily get all the defective genes from your one parent. If you get a working copy from your other parent, you may be functional, may have a functional version of that gene. So we'll talk more about this again when we get to genetics, and we'll start learning about what it means to be dominant or recessive and things like that. So, but that's really why it is. So you have two copies of every chromosome, which means you have two copies of every gene. So here's just for example, this is what a homologous pair might look like if I put some traits on it. So freckles, you might have gotten the freckle version of the gene from your mother, and you might have gotten a no freckle version from your father. Maybe this means you have freckles. You might have gotten a brown eye version from your mother and a blue eye gene from your father. And we'll talk about why this would mean you have brown eyes in the next chapter. You might have gotten blood type A from your mother and blood type O from your father. And we'll talk about again in the next chapter why you'd be blood type A. So you have the ability to pass on freckles or no freckles, brown eyes or blue eyes, blood type A or blood type O to your offspring. Okay. So can sexual reproduction occur this way? You have 46 chromosomes arranged in 23 pairs in your body, and your partner has 46 chromosomes, 23 pairs in their body. If you take a look at this, just to pause before I ask this question again, on the left, this is what's called a karyotype. It's a picture of all the chromosomes from a male. and on the right, it's a karyotype of a female. Do you know how I know that? Okay, yeah, so 22 of these chromosomes are just numbered. The last pair of chromosomes have letters next to them. There's X, Y, and then there's X. The Y chromosome, so if you look at pictures of X and Y chromosomes, the X chromosome will be like this, and the Y chromosome is like that, really short and stubby. What's found on a Y chromosome is only a very few genes, and they basically are genes that lead to maleness. So they're genes that are active during early development of an embryo that turn on pathways on other genes, and we won't get into the specifics of that, that lead to the person being male. So without a Y chromosome, which is the case here, the default condition for humans is female. So female is kind of the default state, and maleness only occurs because of these few genes. So we'll later talk about a little bit about XX and XY, but just for now, XY is male, XX is female. Okay, so does sexual reproduction occur like this? Does a cell from a male with 46 chromosomes and a cell from a female with 46 chromosomes combine to make the offspring? Why do you think that couldn't happen that way? Okay, how many chromosomes would their offspring have?
46 pairs.
Yeah, they'd have 46 pairs, so they'd have 92 chromosomes, right? So that's too many, right? So this is not how it works. So you don't just take any cell from your body and any cell from your partner's body and combine them to make an offspring. That just wouldn't work. Instead, you need to, so this doesn't work, you need to cut the number of chromosomes in half. So you have 23 pairs of chromosomes. You need to make a cell that has 23 chromosomes in it, and your partner makes a cell that has 23 chromosomes. Then when your cells combine, they end up with a cell that has 23 pairs, so 46 total chromosomes. Okay? So the chromosome number needs to be reduced. And by doing that, you're only going to pass on one of those two copies. So that one homologous pair that had freckles and no freckles, brown eyes and blue eyes, blood type A, blood type O, you're either going to pass on the chromosome that has freckles and brown eyes and blood type A, or the one that had no freckles and blue eyes and blood type O, if I remember those correctly. So you're going to pass on only one of those, not both of those, to your offspring. And that's going to be determined when you create this cell. So males, again for here, males are going to pass on only one of these two. Females are going to pass on only one of these two. So who determines the gender of the offspring? Why? Okay, yeah. So during this process, which we're about to talk about, called meiosis, the number of these chromosomes is going to be cut in half. So the female, it's always going to be an X that they pass on. The male, it could either be an X or a Y. If they pass on the X, they're going to have a daughter. If they pass on the Y, they're going to have a son. So gender is determined by the male parent, by which chromosome he passes on. So if you've ever historically heard about, Henry V, who would behead all his wives for not having sons, it was not their problem. It was definitely his problem, right? Okay. So this process of reducing chromosome number, this is called meiosis. So it's a very similar, easy to confuse word to mitosis, but meiosis is different. So meiosis, M-E-I-O-S-I-S, is the process by which you're going to reduce the number of chromosomes. Okay. So in humans, do you know what cells are produced through meiosis? Yep, and that's the only cells in humans that are produced through meiosis. So sperm in males and egg cells in females. So this is what human life cycle looks like. You've got adult males and females, they're going to have some cells in their body, so only cells in their reproductive parts of their body, are going to undergo meiosis. In females, they're going to produce an egg, and in males, they're going to produce sperm. Sperm cell and an egg cell each have 23 chromosomes. Those cells are going to come together in a process called fertilization to make what's called a zygote that now has 46 total chromosomes. Those cells are going to go through mitosis to make an adult human again. So meiosis creates sperm and egg cells. When sperm and egg cells combine, they produce a cell that then goes through mitosis to create an adult. Okay. There's some words we have for these different cells. So the pink cells, the sperm and egg cells, are what are called haploid. And the blue cell, the zygote and the adults here, their cells are what are called diploid. And basically, that's just a measure of how many copies of each chromosome do they have. So a diploid cell, di, meaning 2, has 2 copies of each cell. So we say 2n, so it's 2 times the number of chromosomes. So we have 46 total chromosomes. The egg and sperm are haploid. Hap could kind of like half. It has only 1 copy of each chromosome. So it's got a number of n, which is 23. So in, what is it? I forget these numbers now, but I think it's beavers. Beavers have 24. So they have 48. Sorry, I gave away the answer, but they have 48 chromosomes. So their sperm cells would have how many chromosomes? So if the adult cells have 48, yeah, their sperm cells would have 24. Turkeys have something like 70, I think, chromosomes. And so their sperm and egg cells would have 35. Right? So a lot of organisms are like this. They all have different numbers of chromosomes. In humans, the diploid number is 46. The haploid number is 23. Okay? So the process of meiosis is going to create haploid cells, or cells with only 1 copy of each chromosome, from diploid cells. So meiosis takes diploid to haploid. And what does mitosis do? Diploid to what? So mitosis, you start with diploid. What do you end up with after mitosis? Yeah, multicellular, but they're still diploid, right? Yeah. So this is one of the big differences. So big part of this chapter is comparing mitosis to meiosis. Mitosis, you're going to have diploid cells to diploid cells. You're making copies. Meiosis, diploid to haploid. The process of going from haploid to diploid, that's called fertilization, and we're not really going to focus on that. Okay, so meiosis is going to occur in 2 stages. First thing that happens is sometime during interphase before meiosis, just like in mitosis, your chromosomes are replicated. So you go from having single chromosomes to replicated chromosomes. So a single chromosome to a replicated chromosome, okay? Then, during the first part of meiosis, homologous chromosomes, remember those pairs of chromosomes, are going to line up and separate so that 1 copy of each chromosome goes to one cell and another copy of each chromosome goes to the other cell. So this is what's called meiosis 1, and meiosis 1 is going to separate our homologous chromosomes. So homologous chromosomes separate, that's meiosis 1. So if we started with 46 chromosomes in humans at the start of meiosis, how many chromosomes will be in each cell after meiosis 1? Started with 46. Well, what happened here? We started with 2. How many are in each cell at the end of meiosis 1?
4.
Well, think about that one blue X as one chromosome and the one red X as one chromosome. So if we started with 2, how many are in each cell? Started with 1 red and 1 blue. Yeah, so we started with 1 red and 1 blue, and we have now 1 red in one cell and 1 blue in the other cell. So, yeah, so if we started with 46, we would now have 23. So you've already accomplished your task of cutting the number of chromosomes in half during meiosis 1. I'm going to go into a little more detail about this in a slide or two. So the task of going from haploid or diploid to haploid occurs in meiosis 1. So we have haploid cells now at the end of meiosis 1. During meiosis 2, what's going to happen is those sister chromatids are going to separate, and we're going to end up with single chromosomes rather than replicated chromosomes. So meiosis 1, homologous chromosomes separate. Meiosis 2, your sister chromatids will separate. So at the end of meiosis, you've gone from 1 cell to 4, and you've cut the number of chromosomes in half. Okay. So just to go into a little more detail about this, and this is the big picture. Meiosis occurs in 2 stages. Meiosis 1, where the goal of meiosis 1 is to separate homologous chromosomes. So your homologous chromosomes are going to line up and separate, cutting the total number of chromosomes in half. So what if I started with a cell with 40 chromosomes? At the end of meiosis 1, how many chromosomes would be in each cell? Be 20, right? So you're always cutting the number in half. So meiosis 1 cuts the number of chromosomes in half. Meiosis 2, the goal of meiosis 2 is to separate the sister chromatids, yielding 4 haploid cells. So meiosis 1, you end up with 2 haploid cells, but there's still sister chromatids for each chromosome. And then meiosis 2, you're separating those sister chromatids. So if I could illustrate this, let me see if I can get some. Okay. So let's say I have a cell that just has 2 chromosomes in it. It's going through interphase prior to meiosis. In interphase, these chromosomes are going to replicate, and I now have 2 chromosomes still, but each chromosome has 2 chromatids. Then my cell goes through meiosis 1. These homologous chromosomes will pair up and separate. So now I'll have one cell that has 1 chromosome with 2 chromatids, and I'll have another cell that has 1 chromosome with 2 chromatids. During meiosis 2, within this cell, the chromatids will separate, and I'll end up with 2 cells, each with 1 chromosome that is not replicated anymore. And then this other cell is doing the same thing, separating the chromatids, and so now I have 4 cells, each with 1 chromosome. So I started off with 1 cell with 2 chromosomes. I end up with 4 cells with 1 chromosome each. So I've cut the number in half. So in humans, we start with 1 cell with 46 chromosomes. At the end of both meiosis stages, we're going to end up with 4 cells, each with 23 chromosomes. You guys following me so far? Okay. I'm going to show you pictures of this again. It's not overly complex, but it's a little confusing at first because there's a lot of the same words coming on. We have the same 4 stages: prophase, metaphase, anaphase, and telophase. But because it happens twice, we have prophase 1, metaphase 1, anaphase 1, telophase 1. Then we have prophase 2, metaphase 2, anaphase 2, and telophase 2. So meiosis 1, this is what happens. So this picture here has a cell, a hypothetical cell with 4 chromosomes arranged in 2 homologous pairs. So it has 4 total chromosomes, 2 blue ones, 2 red ones. Prophase 1, you're going to get your homologous chromosomes pairing. So chromosome 1 and chromosome 1 are going to pair up. Chromosome 2 and chromosome 2 are going to pair up. So for us, that would be 23 chromosome pairs. Metaphase 1, the homologous chromosomes are going to line up on the metaphase plate so that one member of the pair is on one side of the cell, the other is on the other side. Anaphase 1, the homologous chromosomes are going to separate. They still have sister chromatids, but their homologous chromosomes are going to separate. And then telophase and cytokinesis 1, you're going to have separation into 2 cells, each of which has half the number of chromosomes and no longer has homologous pairs because you separated the homologous pairs, so it only has 1 copy of each chromosome now. But the chromosomes are still replicated. So this is a replicated chromosome versus a non-replicated chromosome. So you still have replicated chromosomes, but you have half the number of chromosomes you started with. Okay. So you have 2 cells, each of which has half the number of chromosomes, so each of which is already haploid. Each of these cells then will enter meiosis II, and meiosis II is going to separate those sister chromatids. So these 2 halves of each chromosome are going to separate. So prophase 1, followed by metaphase 1, they line up along the metaphase plate. Anaphase 1, the sister chromatids separate. Anaphase 2, telophase 2 and cytokinesis 2, you now have 4 cells, each of which has single chromosomes in it. Okay, so what separated during meiosis I? Yep, her homologous chromosomes. And then what did we separate in meiosis II? Sister chromatids. So that's, if you take nothing else away, those are the big take-homes. Meiosis I separates homologous chromosomes. Meiosis II separates sister chromatids. Where did you first see a haploid cell? A cell with only half the number of chromosomes. Which stage? At the end of? Yeah, the end of stage 1. So the end, so telophase 1 of meiosis I, that's the first time you have haploid cells. So exactly right. So the end of telophase 2, you still have haploid cells. When are you going to get a diploid cell again? Nope. So what did we produce? What do we have 4 of now? So let's say this was a male. What did we make 4 of? We made 4 haploid cells, which are what? What do we call those cells? The haploid cells in a male? Sperms. Yeah, so we made 4 sperm cells. So what's going to happen with those cells? All right, they're going to look for an egg, and fertilization will lead to a diploid cell again. So they're haploid until they meet an egg. Right? So if this was a female, you'd produce 4 egg cells. In females, this happens on average for most females once a month. They'll do 1 round of meiosis, which theoretically creates 4 eggs, but 3 of them become something called a polar body. If you go take developmental biology, you'll learn a little more about this, but only 1 of them actually becomes an egg each month. Granted, some women do this with both of their ovaries, in which case they can produce 2, and that's how you can get things like fraternal twins. But so females do this once a month. Males are constantly doing this, so you have millions of sperm cells. There's a little bit less investment in sperm cells than there are in egg cells. Okay. So, but that's all we're producing now. We're producing either sperm cells or egg cells through the process of meiosis. Every single other cell in your body is produced through mitosis. The only cells you produce through meiosis are sperm and egg cells. So mitosis is much more common in humans than meiosis. Meiosis is just to make reproductive cells. Okay, questions on this? Yep.
Why is it that a woman, as she gets older, her egg cells are like, after 35, they're not as good, and then a man,
Doesn't seem to change much. Well, you know, it's partially that's a matter of numbers. So this process has to happen to produce sperm or egg cells. And this process has to happen properly to produce viable sperm and egg cells. And when you're young, this happens pretty easily. As you age, your body gets less and less proficient at doing it. So it's not a practice makes perfect kind of thing. As you age, the machinery for having this happen doesn't work as well. And so the chances of making a mistake is higher. And if something happens like, and this is something you again would talk about more in 1010, but let's say your homologous chromosomes, rather than them splitting, both of them go to one instead of the other. That happens. It's called non-disjunction. Most chromosomes, if that happened to them and you passed on, this was the egg that got fertilized, it would not survive. It would not implant. You would not get pregnant. It would be aborted before it started dividing even, really, or within the first few stages. Before you even knew you were pregnant, it would be gone. Right? So you wouldn't notice. If you got the other one as well, it's missing a chromosome, so it's not going to work. There's some exceptions. Chromosome 23, that's survivable. You can get, that's called Down syndrome. And that is why Down syndrome is more prevalent with older parents, older mothers usually, because that's more likely to happen. Right? Most other times that would be fatal. It would not survive. Any other thing too that happens during this that's a mistake, usually it means that egg is not viable. In males, you're not just producing one sperm per month. You're producing millions of them. So chances are some of them are okay. And even though percentage-wise, the percentage of good sperm is going to decrease as a man ages, it's still way more than he needs. He only needs one that's functioning. And most of the ones that are non-functioning won't actually go anywhere. They'll be non-functioning in a way that they're not going to swim. Or you might even not even know. It might take you longer to get pregnant, but it'll still work eventually, right? Yeah.
Aren't like females are born with like 1 million egg cells, right? Or something like that.
That's
Like,
the.
my question is like, do they all have like the full amount of chromosomes, and then they have to go through the meiosis or whatever before they can be like fertilized?
The kind of historical thought on that was that during early development in females, in the cells in their ovary, the cells get to a certain point and stop. So they stop prior to meiosis, but you have these precursor cells, and you have a certain number of those. And it's a lot, but it's a certain number. So then basically they're frozen in time. They're not dividing just prior to meiosis, and then each month a follicle develops that allows one of those to start developing. And this is if you take fertility drugs, you can stimulate 30 or 35 of them at a time to develop at once. But they're all there ready to go.
And they all have the full amount of chromosomes.
Yeah, so they're all still diploid because they haven't gone through meiosis, but they're these kind of they're called precursor cells. And they're set for that. There's some debate now, though, on this, because they found in mice, a lot of our knowledge of human anatomy starts with knowledge of mice anatomy, and they're finding that there are stem cells in mice ovaries that can produce more eggs later in life. So it might not be true that it's, you have all the eggs you're ever going to produce at once. It's still something that's commonly thought, but it may not be true. That's something that's still under research. There's some new research being done on that. But yeah, that's the theory, is that they're just there, but they haven't gone through meiosis yet. But they're the cells that are ready to go through meiosis. Yeah. 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 Well, it's good knowledge. It's good things for everybody to know about. So it's the process of life, right? And it's way more complicated, actually, than you'd think. So most people think, well, we're just going to have sex and it's going to work, and we're going to have children whenever we want. But there's a lot of complications, and there's a lot of things that can go wrong, especially as you get older and things change, and there can be problems with that. Yeah?
So if you start off with like 1 million, and then
Yeah.
you get rid of one each month,
Yeah, you have it's more than most women will produce before they go through menopause, which starts shutting down the process of going through meiosis once a month. So that's not usually a limiting factor for women. So, because your body you don't go through menopause when you run out of eggs. There's other things that trigger it that lead to kind of the shutting down of the menstrual cycle, things like that. So, I don't know. I can only tell you guys so much too. So we're reaching the limit of my knowledge of reproductive, but I learn more and more every year. So but yeah, as my knowledge, yeah, you're probably not going to do that unless you're like taking fertility drugs to like overstimulate your follicles every month for years. You're probably not going to run out of eggs. So I would say, don't quote me on that though. It may be possible. I don't know. Okay. Any other questions? This is a great topic because it's really interesting for most people, right? So, yeah. All right. So do you have an understanding of meiosis a little bit? So meiosis cuts the number of chromosomes in half. It does it by separating those pairs, those homologous pairs. Right? This is going to lead us into genetics really well. So kind of before, I am going to start a little bit on genetics today because it's a pretty big topic and I want to spend some time doing worksheets. We may not get to a worksheet today. I'll probably save it for Thursday so we have some in-class time to work on things. But one thing that's going to lead us into an understanding of genetics is understanding all the variety of things that can happen during meiosis that lead to diversity in offspring. So when your homologous chromosomes separate, they line up during metaphase and the homologous chromosomes separate. So you're going to produce two cells at the end. It's just as likely that this chromosome goes to this cell and this chromosome goes to that cell as it is for them to do the opposite thing, for the blue one to go this way and the black one to go that way. When you're only talking about two chromosomes, it doesn't matter. But when you're talking about more chromosomes, it does matter. So here's a cell that has 3 chromosomes, and there's 4 alternatives here. So alternative 1, all the blue chromosomes are on top, all the red ones are on bottom. Alternative 2, blue 1 and 2 are on top, but red 3 is on top. Alternative 3, blue 1 and 3 are on top, but red 2 is on top. And alternative 4, blue 1 is on top, but red 2 and 3 are on top. So now when they go through meiosis, alternative 1 produced half of the cells, half of the sperm or half of the eggs had all blue, and half of the eggs had all red. If metaphase 1 had occurred like alternative 2, you'd be producing cells that had blue 1 and 2 or red 3 and red 3, or red 1 and 2 and blue 3. Alternative 3 produced blue 1 and blue 3 and red 2 versus red 1 and 3 and blue 2. And alternative 4 had blue 1, red 2 and 3 versus red 1 and blue 2 and 3. So we now have 1, 2, 3, 4, 5, 6, 7, 8 different versions of sperm, or 8 different versions of eggs produced from the same 3 homologous pairs. So imagine how many different varieties there can be with 23 pairs. So 4 chromosomes would give us 16 options. 5 chromosomes would give us 32. It keeps doubling every time you add another chromosome with the number of varieties you can create through this. And there's actually more ways of creating variation than just this, which is called independent assortment, but I don't want to get into that in our class because we cover enough variety of topics. I'll wait till you get into a class like 1010. If you took that, you'd learn about more ways that variety is generated. But because of this random way that homologous chromosomes line up, you can end up with a variety. This is why it isn't like, you have one version, your sibling has another, and if you had a third sibling, they'd look identical to one of the two of you. You can have, 15 siblings, and you can all be unique because there's so many different ways of creating sperm, and then there's so many different ways of creating egg, so there's so many different ways that even with the same two parents, those sperm and egg come together. Yes, you're likely to have a few genes in common, and many genes in common, actually, but you're not going to have all the same genes in common, right? Unless you're an identical twin, but that's the one exception, right? So kind of evidence of this variety is fraternal twins. So fraternal twins have 2 eggs, 2 sperm that come together and make 2 embryos. So they're really just as likely to be similar as any 2 offspring. And there's millions and millions of different combinations that can come together by an egg and a sperm, even from the same parents meeting. So the offspring can look drastically different in fraternal twins, right? There's no reason to assume they'll look alike, right? And you can get boys and girls because the sperm that produced the boy had a Y chromosome, and the sperm that produced an egg had an X chromosome, right? So identical twins, if you don't know this already, how that occurs is it's 1 egg and 1 sperm that come together, and then early on, during the first few cell divisions, that cell splits. And so you actually do have the exact same chromosomes because you have the same egg and the same sperm. So that's a different case where you are genetically identical to someone else, and so you would have similar traits. But fraternal twins is 2 eggs, 2 sperm, separate times. Okay, so diversity is generated largely based on the process of meiosis and the process of lining up those homologous chromosomes during metaphase of meiosis 1. Yeah?
Maybe got my job and they're quadruplets, but 2
Yeah.
of them are twins and the other 2 are twins. So how
Okay.
does that work?
Well, that was 2 eggs, 2 sperm, like this, and then each of those embryos split. Were they a product of,
Like 2 are identical.
yeah.
The other 2 are identical.
So they're,
but all twins.
I would suspect that there was probably fertility treatment involved there, but maybe not. No, that's really, yeah, they were natural. They weren't through artificial insemination or something like that. So, wow, that's like a whole bunch of rare events occurring at once. So, yeah, so that means that the mother released 2 eggs that month, one probably from each ovary. They both got fertilized, and then both of them split, which there is genetic disposition to twinning, to having embryos that split during development. So I guess it's not that far-fetched, but usually that wouldn't run in families that also have a disposition to releasing multiple eggs each month. So that's crazy. But yeah, that would be like this, but then each of those embryos split. So you often get that when you have fertility treatments because you're artificially either implanting multiple eggs or you're stimulating release of multiple eggs, and you end up with a high chance of at least fraternal twins. But cool, that's pretty crazy. Okay, so what we're going to start on for the last bit of class, and we'll spend a lot of time on this. I'll kind of get the introduction done today, and then we'll spend some time doing some practice problems and working through these. Because, honestly, from here on out in this class, I find this part is more interesting. You guys have asked me more questions today than you have the rest of the semester, which is fantastic. I hope you stay interested in this. We're just starting to get into the fields that I studied as a graduate student and what I still do today, which is genetics and evolution and taxonomy, which we'll get into. So genetics is something that's really personally relevant, and I hope that you can find parts in this that are really interesting to you like that. So what genetics is, it's the study of heredity. And heredity is the transmission of traits from one generation to the next. So this is something we all are, I'm sure, at least a little bit interested in. How do I look the way I do? And why do I look different than my sibling that has the same parents, perhaps? Or why do I look similar to a sibling that might only have one of the same parents as me? So why do I share some traits? Why do I have other traits that are different? So a lot of our knowledge of genetics comes from this guy here, Gregor Mendel, who did a lot of work in the late 1800s, so the very end of the 1800s. And he did most of his work in pea plants, studying pea plants. So he was a gardener, he was a monk. He had a pretty crazy life. He was actually very unknown when he was doing this research. And it actually, which is kind of a crazy part of this story, is that his work wasn't really found out about until the early 1900s, almost about 20 years or so after he had died, when a bunch of labs tried to plagiarize. So they all found his work, and they said, ooh, this is really good stuff, and they tried to publish it as their own. And then they all saw each other's papers, because they probably were sent them to review, and then realized that they had been caught. So they then finally acknowledged that he had done all this work. So yeah, so there's some unsavoriness to it, but it's reemerged, and we now know that he learned a whole bunch of stuff about genetics. The crazy thing about this, so DNA and our knowledge of chromosomes, we didn't know very much about it until the mid-1900s, so around 1950. So he was doing his work probably about 80 years before anybody knew anything about DNA. So you could kind of keep that in mind a little bit as we're talking about this. We are going to talk about this from the perspective of where we are now, which is we know about DNA, but I'm going to be telling you about Mendel's experiments, which he figured out all this stuff without even knowing anything about DNA. So what he did, he would take pea plants, and pea plants can be self-pollinated or they can be cross-pollinated. So they have pollen and eggs, and the pollen is like sperm, and you can put the pollen in the flower, you can transfer it to from the stamen to the carpel of the flower, and it can then lead to offspring. And you can do that by taking pollen from one pea plant and crossing it with another one, but you can also do self-crosses, which makes it really nice. You can take them and you can fertilize themselves, so they're not resistant to that, which is why plants are often a really nice thing to do genetics in, because you couldn't do that with mice. You couldn't cross a mouse with itself, because that just wouldn't work. We don't have mice that can fertilize themselves. So he would cross-pollinate plants, and he'd record the offspring that he had. The offspring of his experiments or his crosses are called hybrids, so he'd look at the hybrids, and he had numbers for these. He'd call the parents, he'd call the P generation, the offspring would be the F generation, and he'd have F1 and then he'd have F2. So sometimes he would take his parent plants, he would cross them to get an F1 generation, and then he would cross the F1 with itself to get an F2 generation, and we'll be talking about things like that as well. He looked at a lot of different characteristics in peas, but these are kind of the main ones, and we'll be looking at these throughout this chapter. He looked at flower color, so purple versus white. He looked at position, whether it was axial or whether it was terminal, so at the end of a branch or in the middle of a branch. He looked at seed color, yellow versus green. He looked at seed shape, round versus wrinkled. He looked at pod shape, pod color, stem length. So he had a bunch of these different characteristics that he looked at. And all of them, at least in what he was looking at, had two varieties, which makes this easy to start with. So we'll eventually get into more complex things, like humans can have brown hair or blonde hair or red hair. We can have green eyes or blue eyes or brown eyes. But we're going to start by staying away from things that have 3 or more versions and start with things that just have 2 versions. It's either purple or white. It's either yellow or green. It's either round or wrinkled, because it's going to be complicated enough to start with. Okay, so for Mendel, he started with all these traits because for him as well, he wanted to understand the reason why these things happen, so he started with something simple and then worked his way from there. So one of the early experiments that he did is he took what were called purebred flowers that were purple and purebred flowers that were white. And what purebred means is that if he crossed a purple flower, a purebred purple flower, with a purebred purple flower, he would get purple flowers. So there was no kind of dilution in the genetics there. It was all purple. He knew that they had purple traits. So he crossed a purple flower with a white flower plant. So took the pollen from one and crossed it into the other one. And what he found is that every single offspring of that cross, when he grew them up, they had purple flowers. So all of them had purple flowers. So we thought that was really interesting. I wonder what happens if I self-pollinate the F1s. So we crossed the purple flowers with themselves, expecting probably to find only purple offspring, but instead he found purple offspring and some white offspring, and in a ratio of 3/4 to 1/4. So 3/4 of the F2 generation had purple flowers, and 1/4 of them had white flowers. Okay? So we're going to explore why this is, and that's going to be our first kind of introduction, and that might be as much as we really get to today. Okay. So I want to define a few terms before we try to figure out why. His questions and our questions really are, well, what happened to white? Why did white disappear in the F1 generation? And then why the heck did it come back again? And why was it only 1/4 of the flowers have it? Why did it come back in this 3-to-1 ratio? So those were the questions Mendel had. That's what we're going to have. So there's a few terms that we're going to use a lot throughout this chapter. One is the word phenotype. So phenotype is the expressed characteristic. It's what you see, it's what you can measure, it's what's expressed. So color in this case is the phenotype. The purple flower has the purple phenotype, the white flower has the white phenotype. So phenotype can also be things that you can test for, like blood group. So blood group A versus blood group O versus blood group B versus AB, and we'll talk about those eventually. So those are phenotypes. They're what's expressed. And that's really all that Mendel had to go on at the time, because he didn't know the other aspect of this, which is what phenotype is based on. Phenotypes are based on the genes that lead to those phenotypes. So they're based on those chromosomes. And if we're looking at the chromosome, we're going to talk about something called genotype, the genetic makeup. So there is a gene in flowers that codes for flower color, and it has more than one version. It has a purple version, and it has a white version. So you can have organisms that have 2 copies of the purple version. You can have organisms that would have, or flowers that have 1 copy of purple and 1 copy of white. Or you can have 2 copies of the white. So the reason why 2 copies is homologous chromosomes, right? So you have 2 copies of each chromosome. You could have gotten a purple from both parents. You could have gotten a white from both parents, or you could have gotten a purple from one and a white from the other. If you have 2 of the same, so purple, purple, or purple or white, white, this is called homozygous. So homozygous, so the prefix homo means same, zygous is the same genetic makeup. And if it has 1 of each, this one here, it's called heterozygous. Okay, so this is where the audio cut out from my lectures. I'm going to finish this up. There's just a few more slides that we covered in this class. So I was just talking about homozygous, heterozygous. So homozygous means 2 of the same, heterozygous means 2 different alleles. So Looking back at the experiment that Mendel did, he had a purebred purple flower crossed with a purebred white flower. So a purebred flower, because crossing purple flowers with themselves would always give us a purple flower, we would probably assume that both of the flowers were homozygous. So the purple flower would be homozygous for purple, and the white flower would be homozygous for white. So when you cross them two together, the purple flower could only produce purple gametes, and the white flower could only produce white gametes. So any offspring would get one of their gametes from the purple parent and one from the white parent, and therefore they would all be heterozygous. Considering that all the F1 offspring were purple flowers, this tells us that in a heterozygous individual that has one purple gamete and one white gamete, only the purple is expressed. And this is because, in this case, purple is dominant to white. So we've already talked about the genetic terminology for homozygous and heterozygous. In a heterozygous individual, we're interested in knowing which phenotype will be expressed. And if one of the alleles is dominant, that is the allele that is going to be expressed. So a dominant allele is an allele that is always expressed if it's present, whereas a recessive allele is masked in a heterozygote. So to find out what would happen in the F2 generation, so that would be the cross between the two F1 generations, we're going to be using something called a Punnett square. And this is something we will look at in more depth in future class. So to do a Punnett square, what you do is you take each parent and you determine what gametes that parent can produce. So since each of the F1 parents is heterozygous, having one purple and one white allele, they can either pass on the purple allele or the white allele to their offspring. So you'd put one of the F1 parents on one side of a square and the other F1 parent on the other side of the square. And then you'd fill in the boxes to determine what the potential offspring would have. So here we see a Punnett square with each parent giving either a purple or a white allele. If both parents give the purple allele, then their offspring would be homozygous for purple. If both parents give the white allele, their offspring would be homozygous for white. And if one parent gives purple and one parent gives white, then their offspring would be heterozygous. But we know that since purple is dominant, they would have the purple phenotype. So this gives us a phenotypic ratio of 3 purple to 1 white, or 3